Hello, my name is Jaden Martinez, and today I'll be presenting my project for Module 2 assessment assignment. My presentation is entitled A Study of Four Organelles and Passive Cellular Transport. I hope this presentation provides y'all with information about a few organelles and one transport mechanism that'll help you on future tests or just help improve your general biology knowledge.
Okay, so first I'll be giving a brief overview of what I'll be discussing in today's presentation. First, I'll be discussing four different organelles: the nucleus, ribosomes, cell wall, and chloroplast, and giving their structure and functions. Then I'll be discussing my transport mechanism, which is simple diffusion and osmosis, then applying that to a transport scenario to just further develop our knowledge on simple diffusion. Then I'll be giving a real world application for one of the organelles that I discussed earlier, and then concluding with some summarizing points, as well as providing some questions, and then ending with my references page. Okay, so the first organelle I'll be talking about is the nucleus. The nucleus is the largest organelle in the cell and is the central site for organizing and expressing genetic information. It's where the cell's DNA is stored, and this organelle is commonly referred to as the control center of the cell. This organelle is important to the functioning of the cell because it gives instructions for cellular processes, including protein synthesis, which I'll be discussing in the next slide. For the cell to function, it needs this genetic information encoded in the DNA to figure out what to do, what to make, and when to replicate. This information is communicated via nuclear pores, which you can see in the diagram as represented by kind of those dark circular patches on the outside of the nucleus. These pores are openings in the nuclear envelope, which surrounds and protects the genetic information in the nucleus. Nucleotides are able to travel from the nucleus to the rest of the cell via these nuclear pores, and nucleotides are also able to enter the nucleus via these pores. This organelle is present in plant and animal cells, but is not found in bacteria cells. Instead, genetic information in bacteria are just kind of free floating in the cytoplasm and are exposed to the outside substances and materials that will enter the bacteria cell. And that region is called the nucleoid. So the next organelle I'll be discussing is the ribosome. This organelle is responsible for protein synthesis or polypeptide synthesis, which is very important to the cell as proteins are involved in a whole host of cellular functions such as cell signaling, cell replication, muscle contraction, and immune system functions. Ribosomes are able to make proteins through the translation of messenger RNA strands, also known as mRNA. So the ribosome is structurally a two subunit organelle made of ribosomal RNA and ribosomal proteins. So essentially, the ribosome subunits, one on top and one on bottom, will lock onto a strand of mRNA, as seen in the diagram, and read the codons that are transcribed on the mRNA. At the same time, tRNA will join up and match to the codon sequence on the mRNA. Attached to the tRNA are amino acids, which then will join together with other amino acids to make a polypeptide strand or a protein, as seen in the diagram. So ribosomes exist in bacteria and exist in eukaryotic cells and basically perform the same function, which is to make proteins. However, they vary in size and abundance. Eukaryotic ribosomes tend to be larger than prokaryotic ribosomes, meaning they contain more rRNA and more proteins. So next we are going to be discussing the cell wall. The cell wall is a structure that provides extra support and extra protection to the cell. Similar to the plasma membrane, it is semi-permeable as it allows different substances and molecules to enter and exit the cell. In plant cells, the cell wall is made up of cellulose, glycoproteins, and other polysaccharides. Plant cell walls give plants their shape and structure, allowing them to grow and retain their shape in many different environments. These environments could be hypertonic, hypotonic, and isotonic in nature. Um, which would, of course, affect the amount of water entering and exiting the plant cell. For example, in a hypotonic environment, water would rush into the cell, normally causing the cell to swell up and burst. However, because plant cells have that cell wall, they're able to retain their shape and actually not burst, giving them just extra support. So in the picture, the cell wall, you can see it's surrounding the plant cell, and it's like a darker green color. And again, it just gives everything extra support and protects things from the outside environment. Cell walls are in bacteria, um, and in our plant cells, of course, and in archaea. In bacteria, in bacteria, the cell wall also functions to protect the cell, um, and it also helps the cell not burst. The cell wall in bacteria is made up of peptidoglycans, which are different than the polysaccharides that make up the plant cell wall. Yes, so the next organelle I'll be discussing is the chloroplast, which are green. Anyways, the chloroplast's main function is that they're the site of photosynthesis. Photosynthesis is the process of converting light energy along with other molecules into organic products like glucose and oxygen. Chloroplasts are mainly found in plant and algae, however, they are considered semi autonomous organelles as they contain their own separate genomes that is different than the rest of the genomes in the larger plant cell. Chloroplasts are able to perform photosynthesis due to the presence of chlorophyll and thylakoid lumens. Chlorophyll is a pigment that captures light energy and is what gives plant their green color. The thylakoid lumen are structures within the chloroplast that are key to making ATP and other molecules that are then used to create organic compounds in the Calvin cycle, which takes place in the chloroplast. As you can see here in this picture, the chloroplast is green due to the chlorophyll, but you can also see how the thylakoid lumen stack up on each other within the chloroplast and how they have their own membranes, which is key to those light reaction processes and the Calvin cycle processes. So, chloroplasts are not found in prokaryotic cells, but they share a similar DNA structure to bacterial cells. Because of this, scientists have theorized that chloroplasts evolved from ancient bacteria or ancient prokaryotic organisms and developed an endosymbiotic relationship with ancient eukaryotic cells, and eventually became the chloroplasts that we know them as today. So, moving on, next we'll talk about passive transport. Um, Passive transport is a mechanism that describes how molecules and substances enter and exit a cell. This process can also be described as simple diffusion. In order to exit and enter the cell, molecules must move across the plasma membrane. In simple diffusion, molecules move across this membrane following their concentration gradient, which of course involves moving from areas of high concentration to areas of low concentration. In simple diffusion, a substance will have a higher concentration of that substance outside the cell, where the concentration is lower inside the cell. The substance will then diffuse into the cell, going across the plasma membrane. This process is important as it helps maintain healthy concentrations of molecules inside and outside a cell without requiring the input of energy to move these molecules, like in other transport mechanisms like active facilitation and things like that. An example of simple diffusion is the movement of oxygen into the cell, which is then used for important cellular processes like cellular respiration. In this diagram, you can see this happening as the molecules are moving from areas of high concentration across the membrane into areas of low concentration within the cell. And then an analogy for this process is you could think of it like a concert at Red Rocks, where you don't need a ticket to attend, so you could just walk straight into the concert without any resistance or any barriers, which would be pretty cool. So, related to simple diffusion is a process called osmosis, which again is just another form of passive transport, but it deals specifically with water molecules. So, osmosis is the movement of water from areas of high water concentration to areas of low water concentration, typically across a membrane, following the same principles as simple diffusion. Water can move in and out of a cell via simple diffusion, but often in biological circumstances, water will move via the aid of a channel protein. This channel protein is called an aquaporine, and this protein allows for the rapid movement of water in and out of the cell. This is seen here in the diagram, which shows different concentrations of a solute on both sides of the plasma membrane. Water will move from areas of high water concentration or low solute concentration to areas of low water concentration slash high solute concentration via the aquaporins that span the length of a plasma membrane. This process helps maintain cellular homeostasis by keeping concentrations of solutes at healthy levels for optimal cellular functioning. A real life example is in our own red blood cells, which contain lots of aquaporins, which allow for the movement of water in response to different extracellular solute concentration, which allow the red blood cell to navigate different parts of the body depending on the amount of solutes within the bloodstream. Um, so, going along with the analogy from earlier, so like, say you're going to a concert. Um, And you don't need a ticket, so that there's no real barrier, but you can only enter through certain gates. The gates, of course, representing aquaporins. Okay, so here is a transport mechanism scenario that I came up with related to osmosis. So, what do you think would happen if you put red blood cells into a hypertonic solution? So let's think about it. Think about the properties of, or the principles of osmosis, and then skip ahead to the next slide to see the answer. Okay, so here is the answer to the scenario. So we put red blood cells into the hypertonic solution. We know water moves from areas of high concentration to areas of low concentration. In this case, there's a low concentration of water outside the cell, so water would move via aquaporins out of the red blood cells and into the hypertonic solution. This would cause the red blood cells to shrink and look shriveled if you looked at them under a microscope. Microscope. So yeah, they'd kind of look a little bit like how I've pictured them here in the glass, all shriveled and crumpled up. This process of cell shrinking is called crenation and can actually cause cells to lose their ability to function and cause the cells to die. Yes, okay, so real world connections. Here's a study I found called Plant Cell Inspired Colon Targeted Cargo Delivery Systems with Dual Triggered Release Mechanisms, which was published in 2025 and was based on the plant cell wall. So essentially, the researchers wanted to create a drug delivery system, like a pill, that would mimic the structure of a plant and just provide extra protection to the substances or the drugs inside this pill slash plant cell. Typical pills just dissolve naturally in our body's stomach juices, stomach acids, as well as our small intestines acid, and they dissolve before they reach the large intestine and the colon. This is a problem because there are certain diseases and conditions that are related to the colon. And if drugs can't get there, then the patient can't be treated. So essentially, as you can see in this picture, the scientists devised their own plant cell that mimicked the structure of a cell wall. They put different substances within this pill, basically, and then exposed this pill to different environments that mimicked stomach acid and mimicked colon chemicals, essentially. And what they found was that these plant cells were able to survive the stomach acids because of the cell wall, and then eventually they were able to dissolve and allow their internal components to be released into the colon environment. So again, this could be really helpful for people suffering from conditions like colon cancer or irritable bowel syndrome, known as IBS. Um, just taking inspiration from the cell wall and applying it to medical applications so that people can, again, get the treatment that they need in sustainable and maybe even low cost options. So, yeah, check it out. And so we've reached the conclusion. I believe it's important just to remember that the nucleus is the control center of the cell. The cell wall gives structure to cells like plant cells and bacteria. Ribosomes synthesize proteins through translation of mRNA. Chloroplasts are very important in photosynthesis as they make glucose, which we all use for energy. And then simple diffusion and osmosis are key processes to keeping and maintaining homeostasis within our bodies. Okay, and here are a few questions based on all the topics I've covered, so feel free to pause and answer these questions. Here are my references that I use to cite all the information provided in this presentation. And lastly, thank you for listening to my presentation. I can't wait to watch all of yours, and I hope you have a great day. Bye now.